So the next topic is taxonomy. And as we go through this, we'll cover several different things. We'll cover major groups of microorganisms, but also some of the important tools we need for understanding the groupings. We'll also look at what the taxonomic hierarchy or levels are. So let's get into that first. This is a slide to help you explore the taxonomic groups or taxonomic levels. So here are the taxonomic levels. Species is the second to lowest. Strain is lower than this. Every species is part of a genus. So if we look at us, we are Homo sapiens. Homo is the name of the genus. Sapiens is the species, or the specific epithet. So normally if we're going to name an organism, we say the genus and the species. Like Neanderthal was Homo neanderthalis. Homo floresiensis would be the quote-unquote hobbit found in Indonesia, I think. A genus would be within a family. A family would be, every family is in an order, every order is in a class is in a phylum. And then in the bacteria, we don't have kingdoms, we would skip to domains. So the idea here in this table is that for each of these taxonomic levels, you can see, okay, here is what a human is. We are in the kingdom Animalia. We're in the phylum Chordata, class Mammalia, order Primates. Okay? And so for each of these groups, like within our phylum of the chordates, or the ones with notochords and vertebrae, what's a distant relative? What's the most distant relative I could think of that's still in our phylum? And there are some really weird fish in this group, but I came up with a crocodile and a penguin. They're both in that same phylum and they are distantly related. Another example, in the domain Eukarya, these are all organisms whose cells have nuclei and organelles. And so within that same domain, we have plants like a red oak tree and fungi like a mold. This Aspergillus makes the aflatoxin that gives people liver cancers. Okay, so that's the thing to look at, is as we go into smaller and smaller groups, our distant relatives get closer and closer to us. Like within our genus, that's a fairly small group. A genus is a fairly small group. All the other members of our genus kind of look like us. They're very similar to humans. Within our order, primates, they're all monkey-type things, like lemurs and monkeys. Within our phylum, they all have backbones, but they could be reptiles or birds or fish. And within our domain, they could be really anything that has nuclei. The last part of this table is, when was a common ancestor of this group alive? So for each of these groups, they're made up of, so genus, for example, is made up of several different species that all have one common ancestor. We can trace them all back to a common ancestor. When did that common ancestor live? When do we think that common ancestor was alive? And I don't have all these numbers, but for the genus Homo, it's something like 2 million years ago is when the common ancestor we have with Neanderthals and hobbits, when that common ancestor was alive, something like 2 million years ago. Where the mammals, all of the mammals have a common ancestor. So a platypus that lays eggs, and a kangaroo that raises its offspring in a pouch, and us, we're all mammals, but these are fairly different from us, right? And our common ancestor that we have with them is farther back in the past. You have to look back 300 million years to see the organism whose descendants were as different as the platypus, the kangaroo, and us. And then if we want to go back to the biggest group we have, a domain, Well, the common ancestor that we have with oak trees and molds, so the common ancestor of us, plants, and fungi, you'd have to go back something like 2 billion years to find that common ancestor. So what I want you to know, what I want you to pay attention to is how big are these groups? How different are the distant relatives within these groups? Was the common ancestor recent, or was the common ancestor very ancient? And as you go up or down this list, what are those trends that we see? So make sure you know these taxonomic groups. If you've taken biology and now you will have taken microbiology, you should be able to name these. There's a mnemonic device you can use about Katy Perry that will help you remember this order: domain, kingdom, phylum, class, order, family, genus, species. Okay, enough of that. Okay, so these are all the groups that we'll look at in this class, all the different big taxonomic groups. Bacteria is going to be a big group. Protists and fungi are within the domain Eukarya, and those are groups we'll look at. Archaea are their own domain. We will look at those, and these are all single-celled organisms. There are bigger organisms, little animals called helminths. I won't cover them in this class, but if you look in your textbook, you'll see them. And then we have some subcellular or non-living things we'll cover: viruses and prions. These are not made of cells. These are made of parts of cells. And they don't trace their lineage back in the same way that cellular life traces our lineage back. We don't have common ancestors with viruses. Let's see. Right, so let's think a little bit more about these groups. Eukarya, this is this big domain that we are in that also has protists and fungi. And then we could contrast the eukaryotes that have nuclei and organelles with single cells that are tiny, much smaller, and they don't have nuclei, they don't have organelles. These are bacteria and archaea, two different, very different domains, but under the microscope they look very similar. At the other end we have things like species. So, oh, that should be italicized. Darn. Anyway, this should be italicized. Escherichia coli and Homo sapiens are species. So Escherichia, that's a genus. Homo is a genus. But if you say both the genus and species, you are naming a species. And they both should be italicized. So what is a species in animals or plants? Well, we kind of define that by the fact that they can sexually reproduce. So two members of the same species can reproduce sexually, assuming that they are whatever kind of male, female, positive, negative, whatever that type of organism has. So like fungi don't really have male and female, but they have one type and another that have to get together. Animals typically have something like a male, something like a female that have to get together. But the point is, if they're the same species, they can reproduce. If they're not the same species, they cannot. And so that's kind of an easy way to define a species for animals. But bacteria and archaea don't reproduce sexually. They reproduce asexually. They just split in half to make new cells. So how do we define species? Well, there's no straightforward way to do it. There is no foolproof way to define a species of microorganisms. Likewise, a genus is another difficult thing to define. It's not that easy to define for plants or animals either, because a genus is just a collection of closely related species. So you would find definitions, but people would have to think and come up with a useful definition because there isn't an obvious biological definition. We're going to talk about these a lot, and the plural is genera. So we'll talk about a lot of different genera in this course. So just be ready for that. But what this specific slide is showing you is how to write and how to interpret written names of genus and species. So this is an important thing. So look through this slide, because you should know if you see this is a genus, but this is a species, and this is a species. So work through that, make sure you understand it. Right, so how do we define a species in something like bacteria? We have a phylogenetic species concept in bacteria. You're going to need to take some time, I think, to understand this slide. So let me walk you through it a little bit. The way we talk about this is we say two different bacteria are in the same species if, and we say they're in the same species if they have greater than 97% sequence identity in their 16S ribosomal RNA gene. And then skip this for now. So what that means is, well, sequence identity is the percentage of nucleotides that are identical between them. So if you go back to the phylogeny slides and you saw that example I gave of building a phylogenetic tree, sequence identity would have been the percentage of identical bases when you compare any two of those sequences. And so if two different bacteria have more than 97% sequence identity in their 16S ribosomal RNA genes, we say they are the same species. This is based on our historical taxonomic systems. When we grouped bacteria into species, once we got the ability to sequence DNA and do this kind of more rigorous way of analyzing these relationships, we kind of set this 97% threshold to mimic the idea we already had of bacterial species. So again, if these bacteria have greater than 97% identical 16S ribosomal RNA genes, we say they are in the same species. And they will not have 97% or greater sequence identity with bacteria from a different species. Genus is a little bit harder. We could set that threshold around 95%. And so all this can be done just by sequencing one gene from their genome. Don't worry about DNA hybridization, but that was the official definition for species in the 80s and 90s. And if you're trying to prove to someone that you have discovered a new bacterial species, you would use both of these techniques. So we're going to look at different species, but today we're going to look mostly at big groups of bacteria. We're going to look at the phyla. Remember, the phylum humans are in is the phylum of chordates, which is mostly made up of the vertebrates. So anything with a backbone, or specifically the notochord within the backbone. And so that's kind of how broad phylum is. And so these are bacterial phyla. And the size of these boxes is sort of how many different members we find within these phyla. The biggest group is the proteobacteria. Lots and lots of proteobacteria that we're going to see in this class. You don't have to memorize this diagram. You only have to know these if I go through them on a slide, and I will go through them, some of them. This is our universal phylogeny again, and remember, I'm not going to make you fill in all of these labels or draw this, except in as much as the things I have shown you in these videos. So what this is showing is the group of gram-positive bacteria. And there are really two different phyla within the gram-positives. That's based on a test we do in the lab called the gram stain that can give a positive or negative result. And bacteria from the gram-positive group give a positive result in that test. And I'll show you that in a minute. But the point is that pathogens, so bacteria that can cause disease, about half of them are gram-positive. And so even though gram-positive bacteria make up a small amount of the total different types of bacteria, they are important to us. And that means that the gram stain ends up being an important part of microbiology. So you're going to do a Labster simulation on the gram stain, and I'll show you how it works in the coming slides. Right. So the gram stain is a 4-step process. And this slide is showing you the 4 steps and what the effect would be on 2 different cells, one that's gram-positive and one that's gram-negative. And the idea is we start with cells where we don't know if they're positive and negative, and we do these steps, and at the end, a gram-positive cell would be purple, and a gram-negative would be orange, red, or pink. One of those colors. So under the microscope you could tell by the color whether it's gram-positive or gram-negative. Whoops. These are the steps, and the idea is first we use a purple stain that stains peptidoglycan, and I'll show you what that is in the next lecture. And then we add iodine, which complexes with it and makes it stick. And then we wash it out with alcohol. And the idea is a gram-positive cell will hold on to the stain, and alcohol won't remove it, and a gram-negative will lose the stain. Alcohol will remove it. And then we stain with a pale dye that is orange or pink. And you can't see that orange or pink in the gram-positive because the purple dye is darker, but you can see it in the gram-negative. And that's necessary because if you remove this stain and you don't add the purple stain, this is invisible under the microscope. So we have to add this stain to make gram-negative cells visible. And this is kind of what it would look like under the microscope. Notice how ugly this is. That is what you would expect bacteria to look like in a teaching lab under the microscope. They are so tiny, you have to use the maximum magnification, and that gives you the maximum optical artifacts. So this is a mixture of gram-positive and gram-negative bacteria that have been gram-stained on a microscope slide, and this is what it looks like at the end, where these dark blue gram-positive cells and these pinkish orange gram-negative cells are mixed together. And again, all these steps were done on all those cells to figure out if they're gram-positive or gram-negative. Okay, so I'm going to end this video here, and we'll move into the specific phyla you need to know in the next video.